Hello and welcome. Today, we're diving into a fascinating question. Where did the sugars essential for life, like those in RNA and DNA, come from on early Earth? For a long time, a key suspect has been the Formose reaction, a process known since the 19th century. It's compelling because it can create sugars from formaldehyde, a simple molecule likely present on the young planet. This reaction could, in theory, produce ribose, a crucial component of RNA. However, the classic Formose reaction has some significant problems when we think about actual early Earth conditions. One major issue is its lack of selectivity. Instead of producing a clean set of useful sugars, it generates a complex, tar-like mixture of countless organic compounds. Imagine trying to isolate pure ribose from this Formose tar in meaningful amounts. It's a huge challenge under plausible prebiotic conditions. Moreover, the reaction typically needs high concentrations of formaldehyde and fairly specific alkaline catalysts, conditions perhaps not universally available or stable on primitive Earth. Even if formed, the sugars themselves are rather unstable, especially in the alkaline environments that help create them. Because of these challenges, scientists are actively re-evaluating the Formose reaction and searching for more realistic pathways to life's sugars. Recent research focuses on clever solutions. For instance, minerals common on early Earth, like borate minerals, are being investigated. Borates have been shown to stabilize sugars such as ribose. They could have captured these sugars from the complex mixture, protecting them from breaking down. Borates can also form complexes with sugars, potentially influencing their formation and nudging the Formose reaction to produce more of the biologically vital ones, particularly pentoses like ribose. Beyond improving the conditions, alternative chemical routes are also under the microscope. Scientists are looking closely at glycolaldehyde, an important intermediate in the Formose reaction. Perhaps glycolaldehyde and another simple sugar, glyceraldehyde, were formed through different processes or became concentrated in particular environments, allowing for more controlled sugar synthesis. Some studies even suggest that these precursors might have been generated by the photoreduction of carbon dioxide or other geochemical processes. The quest for biotic sugars doesn't stop there. Researchers are exploring other potential sources entirely. For example, could sulfidic chemistries, like those found in hydrothermal vent environments, have played a role? Or reactions involving cyanide derivatives? Another exciting area is the interplay between early carbohydrate chemistry and other key molecules like amino acids or nucleobases. These interactions might have stabilized or selected specific sugar structures vital for life. This ongoing re-evaluation of the Formose reaction highlights a broader trend in origin of life research. We're moving away from viewing prebiotic chemistry as a set of isolated reactions and more towards understanding it as interconnected chemical systems and networks. The emphasis is now on identifying specific geological settings and chemical scenarios on early Earth that could have promoted the selective production and stabilization of life's essential molecules. So, while the Formose reaction remains a powerful concept for generating sugars from simple starting materials, its precise role in the origin of life is likely more nuanced. It might have operated within specific geochemical niches, or perhaps it worked in tandem with other chemical processes that provided the necessary selectivity and stability for life's building blocks, including those vital sugars, to emerge.